Hi everyone, we might make a start now. I'd like to begin by first acknowledging that the Wurundjeri people of the Kulin Nation are the traditional custodians of the land on which we meet today, and pay my respects to elders past, present and emerging, and extend that respect to any Aboriginal and Torres Strait people here today. Today we're lucky for our postgrad seminar today to be welcoming Dr. Haloom Rafehi, who is a senior research officer and bioinformatician in the Population Health and Immunity Division at WEHI. Her research is focused on the discovery of novel genetic variations that cause rare neurological disorders such as cerebellar ataxias, with particular emphasis on the discovery and characterization of pathogenic repeat expansions. Her work on the genetics of ataxia has led to the discovery of two novel pathogenic repeat expansions that are amongst the most common causes of adult onset ataxia worldwide, and has been pivotal for the improved genetic diagnosis of people living with these disorders.
Thanks, Jacob, for that introduction. So today I'm going to be giving an overview of basically genome analysis in the context of monogenic disorders and how we can use genomics to help improve the lives of people living with these disorders. All right, so I thought I would start with a bit of a recap on genetics and monogenic types of disorders before we go into deep into the genomics. So a monogenic disorder, we typically think of it as being one gene causing a specific disease. So mutations or variations in that gene will cause a specific disease outcome. So this is an example of a pedigree, a family pedigree showing in green individuals who carry a variant, and that variant then causes that disease in the family. These variants can be inherited, or they can also arise de novo, meaning they're newly acquired in that individual or the family. So this is in contrast to polygenic disorders. So I put here a Manhattan plot from a recent Parkinson's disease study. So this is a GWAS. So you might have seen a lot of these types of figures before. So this is very different to what I'll be talking about today. These are polygenic disorders. So this is when you have a high burden of common variants that by themselves don't cause disease, but when you have an excess of these variants, you will develop a disease. So Parkinson's disease is an example of that. However, there are also monogenic causes of Parkinson's disease, although they only represent about 20% of cases. However, you can have monogenic causes and polygenic causes of Parkinson's disease, as an example. So where today we'll be focusing on the monogenic variants. These are the rare variants that if you have this variant, you will, depending on the variant, you will always get the disease, or you will have a really high probability of getting that disease. So why is genetic diagnosis and genetic testing and this type of research important? What difference does it make to know what gene is causing the disease? So it's actually, for the patient, it is very important because it gives them answers. They now definitively know why they have a disease, and then also it provides important clinical information. If you know that your disease is caused by a specific gene, the doctor will be able to give you a better prognosis and offer better treatment options. It also opens the door for reproductive options. If it's a very severe disease, you might want to make sure that you're not passing on that variant to the next generation, and then the only way you can do that is if you know what the variant is. You can also access support groups, so you may have a specific type of disorder, and then you can access a support group that will put you in touch with other people who have that disorder. It also allows you access to research trials, clinical research trials. It gives you information to pass on to your family, so they might want to know, are they at increased risk of developing this disease or not? And also can, really importantly, just offer some relief. And for example, you might have a parent who may feel guilty that they did something to cause this disease in their child, when actually it's nothing to do with what they did. It's just, it's just the genetics. So when it comes to genetic variations that can cause monogenic disease, there are a lot of different types of variations that we can consider, and I'll just give a brief overview of what they are. So probably the one that we think of most often is the point mutations and small indels. So these are when you have a single nucleotide variation that might change into a different base, maybe an A to a G or a T to a C, for example. And then we also have what we call small indels, where you have a deletion. So that will be like a small deletion or a small insertion. But the key point here is we're dealing with only a few base pair changes. And typically these are quite easy to detect with genome sequencing technologies. We then have more complex variations. So for example, structural variations and copy number variations. So these can be very large, they can be very complex, and they're a bit harder to detect. And for example, especially with large duplications or deletions, they can cover, like maybe 10, 20, even 100 genes. So we often see these type of variations in really severe syndromic disorders. And then we also have a subset of variants called repeat expansions of short tandem repeats, and this is my area of expertise. So this is when you have basically a repeated motif of DNA, and we all have hundreds of thousands of these, and they're typically benign. But for a small subset of these genetic variants, when they become expanded, meaning they exceed a certain threshold in terms of length, they can cause disease. All right, so just a bit of a recap on how these variants are inherited in the context of disease. You can have autosomal dominant inheritance. So what this means is that if you have one copy of the variant, you will develop the disease and you can pass that on to your offspring as well. So in this example, a full blue person is someone who carries the variant and also has the disease. Autosomal recessive inheritance, you have to inherit 2 copies from your parents in order to develop the disorder. So if you have just 1 copy, you don't get disease, you have to have 2. And we also have X-linked recessive, where it can be carried on the X chromosome. So in the recessive model here, the female carrier will have 1 copy, they won't develop disease, but they can pass that on to their male offspring who will have 1 copy, and because they only have the 1 X chromosome, they will develop disease. So I just want to briefly talk about cerebellar ataxia, which is the disease that I primarily research. And not that this is important for the context of genomics, but it's just I would like to give some examples of my research to help explain what we actually do and how we can discover different disease variants. So cerebellar ataxia is a neurological disorder that primarily affects gait. So gait is the way we walk, and people who have ataxia have an impaired, what we call an ataxic gait. So they have poor balance and coordination, and ataxic syndromes often present with other symptoms as well, such as nystagmus, which is unusual eye movements, also neuropathy, so their fingers can become numb and their toes can become numb. And it primarily occurs as a result of impaired function of the cerebellum, which is this region of the brain here that controls movement and coordination. And it's thought to be primarily a result of dysfunction of a subset of cells called Purkinje cells. So this is quite a rare neurological disorder. It affects about 1 in 10,000 people, so that might be about 2,000 to 3,000 people in Australia who have the hereditary form of this disease. It's primarily caused by repeat expansions, but it can also be caused by SNPs. So those are single nucleotide polymorphisms, so a single base pair change, or structural variants as well, but that is less common. Okay, so I want to talk a bit about the study design for genomics, because this is really important. You really have to have the study design in place in order to be able to properly do your research. And this is especially important because we're dealing with actual people's DNA, actual patients, so you really need to get this right, especially from an ethics standpoint. So you need to consider your research question and your disease phenotyping. What is the goal of the research that you're doing? Is there benefit to be found here for the patients? And also, is your disease actually well defined? So I'll give an example for all of these points from our ataxia research program to help understand what I mean. So an example of our research goal is to discover new genes that cause ataxia. And to do this, we need to have a really tight definition of what we mean by ataxia, because there's actually many types of ataxia. So for example, we study cerebellar ataxia, but there's also sensory ataxia. And is the ataxia the primary symptom, or is it part of a syndrome? Because ataxia, the symptom itself, can occur as part of other syndromes or other disorders. And is it a genetic ataxia or an acquired ataxia? Because ataxia can also occur as a result of injury or drug abuse, or alcohol abuse, for example. So this is where it's very important that if you're a scientist and you want to study this disease, you need to have a really good team of clinicians on board who understand these points and know how to do the proper phenotyping to make sure that you're recruiting people to your study who actually have the disease that you're interested in. And then it's also very important, of course, that your project is well funded and that the goals of your project align with the budget that you have, and that you have the proper collaborations in place. So I already mentioned working with clinicians, but for example, we also work with molecular scientists. So all the bioinformatics happens here at WeHi, at our team with Melanie Barlow, and the clinical recruitment and the molecular work is done by our collaborators at the MCRI, as well as other neurologists from across Melbourne and also across Australia. And of course, ethics is the most important thing here. You need to have an ethics approval when you're working with actual human patients. And we have that ethics approval done via MCRI and WeHi. And we're in a good position in terms of funding. However, that wasn't always the case. We had to prove that we could do what we do without any specific funding first, before we then were able to get more funding to set up a ataxia research program. And then when it comes to recruitment and consent, this is again really important when dealing with real human participants. So there's many different ways to go about this. You can go via clinicians, individual clinicians, via hospitals or genetic testing centres. Do you go direct to patients? That's a bit more complicated. Are you looking at local, national or international? If you are looking at international recruitment, you need to consider, do you have appropriate ethics in place? Is language barrier and consent going to be an issue? If your collaborators don't have good English, are you able to properly communicate with them and understand that they've got the appropriate consent from their participants for you to be able to do your study? Are you recruiting families, including people who are both affected and unaffected, or are you only recruiting individuals, so one person from each family, and looking at a more cohort-based type of study? So this is all information you need to consider beforehand. And it's also, of course, really important that your consent process has been reviewed by ethics committee to make sure that people joining your research program understand what they're consenting into and what type of research you will be doing. So an example from our program, so we recruit people with ataxia from across Australia, and we do that via neurologists, so they recruit their patients. So the actual clinician is aware of our program, and they will put forward people who they think would be suitable. We mostly recruit just individuals. However, in some specific cases, we will recruit families if there is a large family who has multiple affected individuals. And then the final decision of whether someone is recruited into our cohort is made by our lead ataxia expert, who decides if this individual actually fits into the proper category of the ataxia that we want to study. So the other thing to consider, also again, this is part of the study design that you need to do really early on, is what type of tissue you want to be collecting. Typically we collect whole blood to do whole genome sequencing, but depending on the disorder that you're studying, it might be difficult to actually access whole blood. So for example, I previously worked on an autism study, and it was very difficult for some of the participants to get a blood draw because that's something that they wouldn't really be comfortable with. So in that case, we might do a saliva or a buccal cell swab, which is just a swab inside the cheek. Are you looking at nuclear DNA or cell-free DNA? Cell-free DNA can be really interesting in the context of cancer, for example. Are you doing tumour biopsies? And in that case, do you also need a healthy tissue biopsy for a comparison? Because we know tumour DNAs can be very different to a person's germline DNA. And if you are doing tissue biopsy, is this something that you can collect just from the skin, which is very simple to collect? Or is it something you need to access during surgery or post-mortem? So in the context of our study, we ideally would like to get brain tissue, but obviously that's not something you can easily access. Sometimes for some disorders, there are surgeries that they do on the brain, such as deep brain stimulation, where they can actually access a very small amount of tissue that they can use for their study. So that's something to think about beforehand. You also might want to collect tissue for validation studies. So if you do find a variant that you're interested in, you might want to do further experiments to prove that it's actually pathogenic, and in that case you might want, for example, fibroblasts from a skin biopsy that you can use for stem cell generation and cell differentiation. That's something we do actually do in our study. And then finally, you need to think about how are you storing your genomics data. So is it somewhere that you have sufficient space for all the storage, and is the data safe, and is it protected from people being able to access that data who should not have access to the data? Because remember, this is very sensitive genetic information from people in your study. So in the context of our research, as I said, we use whole blood. It's not possible to get cerebellum, but we do have some people who have expressed interest in donating part of their brain for research. But obviously that is a very long process. And then we also use limited skin biopsies for differentiation models, only in cases where we think that is worthwhile pursuing. And we also have very secure storage at MCRI and WeHi servers. And then finally, we need to consider what type of sequencing are we actually going to do. And this, of course, will also be dependent on your budget. But we have exome sequencing versus whole genome sequencing, where with the whole genome sequencing, we can sequence the person's whole genome or their DNA, versus an exome where we're only looking at sequencing the exons of genes. So I think people have really started moving away from exome sequencing now that whole genome sequencing is much more affordable. And the benefit of whole genome sequencing is it allows you to look at intronic variants as well, which can very often cause disease. And then the other question is, are you doing short versus long read genome sequencing? And I have a slide about that and the pros and cons later on. Of course, the biggest con for long read sequencing is still very expensive. And then you might want to also think about doing RNA sequencing. Would you be doing bulk RNA sequencing or single-cell RNA-seq? Is that something that you need for your study? And that's going to have an impact on how you actually collect your tissue and how you store your DNA. And then you need to think about validation studies. So do you need extra material to validate your findings, or are you going to generate cell culture models? That's something you need to think about in advance, and also there is potential to do animal studies. So in the context of our research, we're funded for 800 short reads whole genome sequencing samples. So that's quite a lot. We really couldn't do that with that number with long read sequencing due to the cost. And we also do work on neuronal differentiation models with fibroblasts. Okay, so when it comes to genomic sequencing, you need to think about whether you're going to use short or long read sequencing. So just to recap, short read sequencing, your reads are 150 base pairs in length and they're paired. So you have a long fragment of DNA and you're sequencing the start and the end of that fragment. This leads to a very patchy alignment. You're going to have regions of the genome where you can't align properly because they might be repetitive elements, for example, and you can't uniquely map that read back to a specific location in the genome. So that, in theory, is solved by long read sequencing, where you have these really long reads that are much easier to align back to the reference genome because they can be, tens of kilobases in length. So they're really much, much larger. However, there's pros and cons to both, and I'll just go through that quickly. So we still do short read sequencing as our main type of sequencing, and the reason for that is, first of all, it's a well-established technology. The quality is really good. It's cheaper. It offers better coverage for the amount that you're spending. It has very high accuracy in terms of base calling. It's easily accessible. There are many providers, and prices can be competitive because of that. And there's very well-established pipelines for analysis. One of the main cons, of course, is that short-read sequencing can be difficult to use for detecting large and complex structural variants. It's not impossible, it is difficult, and I will show examples of that later on, of how that can be overcome. But that is one of the main reasons why you would want to consider long-read sequencing. So again, the pros for long-read sequencing are that it's you have long reads, of course, so that's good. It's better for detecting large and complex structural variants. However, it is expensive. It is a newer technology. So it is rapidly improving, and it's improved a lot over the last few years, but it's still new and developing. The cost to get high coverage is still quite high, and it's not as widely available. So there aren't as many providers who you can go to get competitive pricing when you're looking at long-read sequencing. And actually, one thing I forgot to mention is databases. We rely a lot on databases where they've sequenced hundreds of thousands of people so that we can compare our results to those results. And all databases at the moment really are primarily made up of exome and genome short-read sequencing. We don't have the equivalent for long-read sequencing to be able to compare back to say, this is a variant we can only detect in long-read sequencing, but how do we know if it's rare or something that's common? And we just don't know it's common because we don't have a large enough population database from long-read sequencing to say this is common. So once you get your data, you need to think about how you're going to actually process the data. And when it comes to short-read sequencing, there are very well-established guidelines and pipelines that you can use. So we use something called the GATK best practice, and this is a published guidelines. There's a really great website for that, and I've got a list of resources at the end of my talk that you can follow up if there's anything you're interested in. So this is an example of an Xflow pipeline called SARIC, which you can implement yourself, and they've put together a pipeline based on the GATK best practice guidelines that will do all your alignment and variant calling, as well as some structural variant calling for you. So this is a really great tool. We personally don't use that. We have a fantastic RA, now PhD student in our lab, who's developed an equivalent pipeline that is custom tailored to our needs in our lab, but it's very similar in terms of concept. So I just want to highlight really quickly some key file formats that are really important for the type of research we do. So we have the FASTQ files. These are our raw sequence data that we get straight off the machine. We then need to do a process called alignment, where we figure out where those reads come from in the genome, and that gives us files called BAMs or CRAMs. CRAMs are very similar to BAMs, they're just smaller size, because we're dealing with quite large file sizes here, so it's always good to save some space where you can. So basically, this is a file that lists where your read maps to in the genome and quality scores, and how well it maps. And then we have VCF files. So this is a text file with a variant call. Basically, it tells you this person has this variant in this region of the genome. So you can imagine these are quite large files when you're dealing with genome-wide data. And the standard VCF that you would get out of this type of pipeline is really just looking at SNPs and small indels. So indels means insertions, deletions. It's not really looking at the large structural variants or repeat expansions. Okay, so I'm going to go through a couple of case studies from our research to highlight what this pipeline would look like in practice. So this is an example of a family with ataxia that we started studying in 2014, which is actually about 3 years before I joined Melanie's group. So this family goes by the A1 label, that just means ataxia 1, it's our first family, who has ataxia. So you can see from this pedigree, there's quite a few people affected in the family. So the black squares are people who are fully affected, and the grey squares are people who are mildly affected. And we have genome sequencing data on the individuals who have the circle around them. So individual 3, 2, and also 2. So they're in different generations, so that's why the numbers are like that. So we have data from quite a few people. So we've done our whole genome sequencing, so then what? We've just got a list of variants, so we need to start digging to see how do we actually find the variant in this family that's causing disease. So the first step is something called linkage analysis, which is something you can do in family studies. So the idea here is to take the genomic variants from our 3 individuals in this family and run them through a tool called LinkDataGen, which applies many different tools. And in simple terms, what this does is it takes the people in the family who have the disease and the people in the family who don't have the disease, and then it compares their DNA and it says, the people who have the disease all share this region of the genome, and these regions are not shared by the people who don't have the disease. So really, we're looking for something. In this family, we've applied a dominant inheritance model, so they only need 1 allele, and we've applied what we call a fully penetrant model. So penetrance means basically, in a fully penetrant model, if you have the variant, you must develop the disease. Not all variants are fully penetrant, so in some cases you might want to apply a model where you're doing what we call incomplete penetrance, and you might specify that the variant only causes disease in 50% of cases, for example. But in our situation, we are assuming a fully penetrant variant, and we identify 3 regions of the genome with a LOD score of about 2. So LOD scores need to be 3 and above to be considered significant. However, I think the reason we didn't really get significance is our study was a bit underpowered, so we would have needed more people to put into our model to be able to say, to identify just one region of the genome as high confidence, this is where our variant is. So we identified 3 regions, and what we noticed was one of these regions on chromosome 2 is actually listed on a website called OMIM, where they list known diseases. And it's listed as SCAR25, so that stands for spinocerebellar ataxia 25. And it's listed as an unsolved disorder, and they have examples of a French ataxia family who also have linkage to this region. So immediately we're thinking, this is really interesting. This looks like this family must also be SCAR25 if they have linkage to the same region. So this is before I joined the project. What I understand is one of our clinicians reached out to the French research team who was working on this family to see if they were interested in collaborating, and unfortunately they were not. So we had to go at it alone. So what we start off with is variant curation. So what does this mean? Well, we know that we need to be looking at chromosome 2, so we can filter variants for only those in our chromosome 2 linkage region. And we're filtering for variants that are shared by all affected family members, so it must be present to cause disease. And then we also need to know, is this a rare variant or just a common variant that we're not really interested in? And we use a tool called, a resource called Nomad, and I'll talk about that a bit later. So Nomad has basically profiled hundreds of thousands of individuals, so we're looking for something that is either very, very rare in Nomad or completely absent in Nomad. And we also have tools that will predict, is the variant pathogenic or not? Because we need to know, because we do end up with a lot of variants, so we need to know, is this variant predicted to be pathogenic? And then of course, is it expressed in brain? Because in the context of our disease, a neurological disorder, it needs to be expressed in brain to be able to cause disease. So once we applied all our filters, we are left with this single candidate variant on chromosome 2 called PNPT1. So this is a summary output of the different parameters that we look at. So what we can see is that this is a splicing variant. SIFT, polyphen2, and CAD are pathogenic scores, but because this is a splicing variant, it doesn't get a score. So we don't have scores here. And we can see from this, this is an IGV screenshot. I'll show a bit more detail later on that, but it basically shows what the reads look like. And from the reads, we can see that the variant in blue is present in all 4 affected people in this family. And the reads look like this in quality, so this is not a false positive call. However, one small problem, this gene is already associated with 2 different diseases in an autosomal recessive fashion. So one of them is oxidative phosphorylation deficiency and another is deafness. So this doesn't really fit what we would expect for a dominantly inherited ataxia gene. And also the splicing variant itself, when we had a closer look, it is a non-canonical splicing variant. So what that means is it's not really in a place where you can say this is very likely to be a splicing variant. And the fact that it's associated with recessive disease already, we kind of went, this is probably just an incidental finding, a variant that they shared probably doesn't cause disease. So just to recap some of the resources and kind of explain the resources that I talked about. So the first one, Nomad, is an amazing resource for the type of work that we do. So they now have 800,000 exomes and genomes that they have profiled and summarized on their website. And although this is primarily a European dominant database, so about 600,000 of these individuals are European. They also have about 150 or so thousand individuals from diverse backgrounds. So you can see different Asian populations, Middle Eastern, African, etc. And the reason that this is really, really important is that different variants might have different population frequencies in different ethnicities. So it's really important that you get a good representation from different groups. So although our research is primarily focused, because it's an Australian cohort, most people in our study would be European background, but if you had a cohort or a study that you were running on a different population, you would expect to see a lot of variance in that population that are not present or not common in Europeans, just because that's how the genetic works. Different population groups will have different variants at different frequencies. And if you find a variant in, let's say, a Sudanese family, and you're like, okay, this is not present in any Europeans, but that doesn't mean it's necessarily pathogenic, because it might be common, or maybe at 10% in Sudanese individuals. So you really need to be able to have a reference population you can go back to from that population to say this is just a common variant in that population. So the numbers are really improving, but obviously we still need a lot more representation from different ethnicities going forward. This is an example of what NOMAD looks like. If you click on a single variant, it shows you the number of allele counts for that variant in different population groups, and how many people were tested from. So this is just a random example. I couldn't show the PNPT1 variant because it's actually 0 in NOMAD, so there's no table for that. But this is another variant where you can see it's not present in any population except for South Asians, 26 alleles out of about 64,000, and one person from a Middle Eastern background and one person from the group is called remaining, so those people aren't really labelled what the ethnicity is. Oh, I forgot to mention this on the initial slide, but another really important research for us is GTEx. So this is a gene expression database from different tissues in the human body, and you can see these ones in yellow over here. These represent different regions of the brain, and in particular, let me see, yeah, this one here, this is expression in the cerebellum. So because we're doing a study where the cerebellum is where the phenotype originates from, or the injury, we need to make sure our gene is actually expressed in the cerebellum. But they have tissues across all different body types that you can reference to depending on what disease you're studying. Another really good, actually I should mention, they also have single cell expression, they have transcript level expression, they have single cell EQTLs, so this is a really fantastic resource, highly recommend. And this is all human data, by the way, not ours. Okay, so another thing that we can do when trying to figure out, is this variant causing disease, is the gene causing disease, is we can have a look and see if someone has done a mouse knockout model. And this is a really good resource for that, MGI, I think Mouse Genome Informatics. So this is the page for PNPT1. So there are about, I think, 3 different animal models here that they've tested, and none of them actually have a neurological phenotype. So again, this is kind of saying, this is probably not worth pursuing this variant in PNPT1. Although I wouldn't rely 100% on mouse models, it's good to get information from them, but I wouldn't rule something out purely based on whether or not there's a phenotype in mouse. Okay, so what next? So I started working on this family in about 2018 after I joined Melanie's lab. So at that point, the PNPT1 variant had already been discovered and dismissed. And as I mentioned earlier, ataxia is commonly caused by repeat expansions and can also be caused by structural variants. So that's what I did for about 2 years. Not full-time, obviously, but just as one of my projects. So I did structural variant analysis, we did repeat expansion analysis, we went back and did bulk RNA sequencing to see if we can find a change in gene expression that might tell us which gene we need to be looking at. We did DNA methylation, nothing, we didn't find anything. So we were really stumped, we couldn't figure this out until we got an email from the French group, and they told us they had variants in PNPT1 in their SCAR25 family. So if they had sent that email about 2 years earlier, we would have not spent so much time doing all this work. But we finally said, okay, it is PNPT1. So we worked together with the French team. So this is the variant, which we didn't think was very promising at the time. And this is a very common story, I should say, in genomics. You have a variant, it is the answer, you just don't know it, and it can be very hard to prove. So in this case, we didn't figure it out until we had another family who also had a variant in that disease causing the same phenotype. So we worked together with that team and we published this paper. At the top, you can see the pedigree from the French family and the Australian family at the bottom. And they were able to figure out basically these splicing variants they had to do molecular work to follow this up. And that's one of the things our team had to consider is, doing the follow-up work. It's very labor intensive, it costs money, it takes time. We didn't have enough information at the time to say it's worth it to go and do these molecular work to show that the variant is pathogenic or has some kind of effect on the protein, which is, again, one of the reasons why we missed it. So, they were able to show in this study that the splicing variants actually were truncating the protein and then causing disease. One thing I actually picked up when I was putting my slides together, and I don't think this was available at the time we did the research, but there's another mouse study website, so IMPC, sorry. I forget what that stands for, but I've got it listed in the resources at the end of my talk. So PNPT1 is profiled here, and you can see the orange brain. That means they do have neurological phenotype. And I guess you can't read that, but anything to the right of this green line is a significant observation. And I've just highlighted there, they've actually picked up gait abnormality, and that is literally what ataxia is. It's a gait abnormality. So I feel like if we had that information at the time, it would have actually been a really strong green flag for us. Hey, we should follow up on this. But I don't think this was available at the time. But yeah, just to show two different mouse databases, completely different results. So the gait was not listed in the previous MGI website that I showed. Okay, so moving on now, I want to give another example. So that was more of a straightforward example where the discovery part was very easy, but the validation and putting it together part was a bit complex. The other, I guess, genetic variants that we can consider are structure variants and repeat expansions that I mentioned earlier. And these are very hard to detect in short-read sequencing data, but in the last, I would say maybe 7 or so years, there's been a lot of advances in the bioinformatics tools that we can apply to detect these variants in short-read sequencing data. So I'm going to go through another case study. So this is Canvas. This is a type of ataxia. And this was the first study we published on ataxia, and we did all this without funding, and this is a project that was able to allow us to establish ourselves in the field and help get funding for us to do more ataxia research. All right, so just to recap a bit, what are STRs and repeat expansions and how do they cause disease? So this is an example of an STR. So it has repeated DNA units. So for example, that could be a CAG that's repeated over and over again. As I said, there are greater than 200,000 in the human genome. They are predominantly benign. They don't cause disease, no matter how big they get for most cases. So variation is normal. For about 60 repeated STRs that we know of, when they become expanded, they can cause disease. And these are, at the moment, they're all located in genes. So it can be UTR, exons, also introns. They can all cause disease. So when they become expanded beyond the threshold, and that threshold is specific for each specific expansion. So the threshold for exonic STRs, for example, can be as low as 30 repeats. But for an intronic one, it might be as high as 600 or 2,000 repeats. So it varies a lot. And this is really interesting for our team because ataxia is the disease that is predominantly caused by repeat expansions, but also the most common, so repeat expansions are most likely to cause ataxia than any other disease. And interestingly, and we don't know why, repeat expansions almost exclusively cause only neurological disorders. No one's really figured that out yet, though, why that is the case. So when it comes to Canvas, we were very, very fortunate that we collaborate with a fantastic clinician called David Schmulowitz, and here in Melbourne, and he actually defined the Canvas phenotype. So he's the person who first published the papers saying, this is what Canvas is, this is what it looks like, and we had access to a lot of families who have Canvas. So as I mentioned earlier, I guess with the SCAR25 story, the more families you have who have the same variant or variants in the same gene, the easier it gets. So this is a pedigree of one of our larger families as an example. So we call this family Canvas 9. So you can see there are 4 affected offsprings and the parents are unaffected. So what this tells us is that this is probably a recessively inherited disorder if the parents are both unaffected but they have multiple affected offsprings. So they need to inherit 2 copies of the variant to cause the disease. And we had multiple families who had this similar type of recessive inheritance pattern, so we were quite confident we were looking for something recessive here. So we did the linkage analysis and we get, for this family, and we get a very strong peak here, you can see on chromosome 4. And this is genome-wide significant, so we were quite confident this is where we should be looking. But also, when we did linkage analysis on the other families, so we had about 5 families who have canvas, we were able to narrow down the linkage region. They all had the same linkage region on chromosome 4, and we were able to narrow it down to this very small region on chromosome 4 that overlaps with about 3 or 4 genes. So we were really confident this is where we were looking at. The initial work on canvas, so this was before I joined the team, the supervisors on the project were able to get Illumina to do, I think, about 12 or 20 free exomes for us as a collaborative thing. So we had a lot of exome data to analyse, but nothing really came out of it. We couldn't find the variant. So we went back and did whole genome sequencing on 2 individuals with canvas from 2 different families. And when we did that analysis, we did STR analysis on that family because we know STRs are the most common type of variant that can cause ataxia. And we found, so we applied a novel method called expansion hunter de novo that was developed by Melanie in collaboration with people at the time who worked at Illumina. And this was done before the tool was even published. So we were able to find a really interesting repeat expansion in the intron of a gene called RFC1, and that's really important because we didn't have intronic data before when we did exome sequencing, but we now have that with the genome sequencing. So in RFC1, there is a reference STR, so it's AAAAG, so it's shown here in the orange, and it's typically quite short in most people. What we found for the 2 people with canvas is that they had an AAGGG, so this is a different motif. And they had it on both alleles, and it was expanded. And we didn't find this in anyone else in our control population that we were looking at. And these are 2 unrelated individuals, so they're not related. So they both had this. So if we have a closer look at what that looks like in IGV, IGV is a really important tool when it comes to genomics for visualizing what your genetic variants actually looks like and if they look real. So at the bottom here is an example of a control. You can see they match the reference genome, which is over here, the green and the orange. I don't know if you can see that very clearly, but it's 4 A's and a G repeated about 10 or 20 times. And then this is what it looks like in the 2 people who have canvas. So they are mapping to a different motif. They have this AAGGG motif, and we can see that it fits the recessive inheritance pattern. So this could still be a coincidence. We didn't really have proof that this is actually causing the disease, and there are no databases for repeat expansions. So for example, with the SNP that we detected in the SCAR25 family, we had so many resources to look at that specific SNP to say, is it predicted to be pathogenic? How frequent is it in the population? etc. There's so many resources. Especially at the time, there's almost nothing for repeat expansions. So we had to do more digging. So for example, we went back to the exome data, and fortunately for us, exome data, it's not very clean. You get reads that are not actually from exons. So we were able to find for a lot of people in the cohort that had 1 read randomly mapping to across this STR, and we were able to show that all of them who had a read there, they had this motif. They didn't have the reference motif. So, that was really good evidence for us. And then another thing we were able to do was haplotype analysis. So what this means is a haplotype is, you have your repeat expansion or your genetic variant, but you want to know, is this a de novo mutation or did it happen once and it was inherited from a common ancestor? So what you do is you look at the rare SNPs around the mutation or the variant, and you want to see, do the 2 people who have canvas, who we have the data for, do they all have the same haplotype? Do they have these rare SNPs telling us that these unrelated people have inherited this segment of their genome from the same individual, like common ancestor, again, providing more evidence that this would be pathogenic. And the great thing about that is we can do that on the exome data. We don't have to rely on the genome data. So we had about 20 individuals who, well, maybe not 20, maybe about 10 individuals who we were able to show they do have a common haplotype, again, providing evidence that this repeat expansion is shared by all these people with canvas and not really present in controls. And that was enough evidence for our collaborators to go back and do the wet lab work, to do the PCR, etc., to show that the repeat expansion is present in the whole cohort in an inheritance fashion, in a recessive inheritance fashion. So one cool thing we were able to do with this information is in the UCSC Genome browser, which is another amazing resource for genomics. And you have to do this in the HG18 version. It's not available in the more recent genome builds. But we were able to take this highlighted region in blue, which is the shared linkage region in the people, in the different canvas families in our study, so the shared haplotype region. And we were able to show in the Europeans, which I think is the middle one here, that red little mountain, that means that region does not get recombined. Again, showing that there is linkage disequilibrium here. So what that means is this region is inherited as a common haplotype. So the fact that these SNPs are together on this haplotype means it's not a coincidence. There is actual linkage disequilibrium at that region. And interestingly, that linkage disequilibrium is not present in this population here, which I think, I cannot read that label. I think that's an African population, and the bottom one is a Japanese population. So you can see very strong linkage here. And finally, one cool analysis we could do from this is something basically called mutation dating. So if you have a variant inherited on a common haplotype, you can take the size of that haplotype, like how long it is, and determine how long ago that original variant occurred. So we applied this method, and we were able to estimate that this AAGGG repeat expansion arose probably about 25,000 years ago, which is pretty cool to be able to show that. So basically what that means is at the time we did the study, what we were able to conclude was that everyone who had canvas actually had this common ancestor who developed this variant 25,000 years ago, which is pretty cool. However, that's not strictly true because we now know in some populations, I think it was a Pacific Islander population, they actually have a different motif that causes canvas in their population, which would have its own founder effect, which is in itself really interesting. Again, highlighting the importance of looking at different populations. Okay, so just to quickly wrap up, there is so much more to cover in genomics, and I just wanted to highlight this paper if anyone's interested in reading what else we can do for diagnostics, and understanding rare diseases. This is a really nice paper that covers in a lot more detail other methodologies we can use as well. And these are the resources I talked about today. This is not an extensive list, but these are some of the ones that we use the most. I don't know if these slides get shared or anything, but you should be able to follow that up in the recording if you're interested. And I just wanted to acknowledge all the people who've been involved in the ataxia work that I've talked about today. So it's a huge collaborative effort between MCR and WEHI, and also a lot of our international collaborators. I'm happy to take any questions.
All right. Thanks for your talk, Haloom. We do have time for some questions now, and as always, I have chocolates.
Lovely talk. So I know you were, with your examples, talking about how you had some different families to confirm what you'd seen. So how many kind of families do you need for it to be sort of able to be translated clinically eventually? Do you know what I'm trying to say? Like, how much power do you need?
It really depends, right? So for the PNPT1, the SCAR25, we had 2 large families plus 1 extra person, and that was enough as long as you do the follow-up work. So theoretically, we could have published the SCAR25 story ourselves, but we just didn't think at the time it was worth putting all the resources into trying to do the validation. So the validation is where it's really important, because we weren't convinced at the time, and it was going to take a lot of money and resources, which we didn't actually have because the ataxia work was not funded at that time in order to follow up on that. So really the answer is 1 family can be enough. The more, the easier it is, right? So with Canvas, that was a really tricky one, and I made the initial discovery in 2 people from 2 different families, but it took a lot of convincing for us. So we kind of had to just ask so many questions, and we did so much more digging, looking at the haplotypes, looking at the exome sequencing before we decided, okay, this is actually worth us pursuing, this looks real, let's go do the validation. So the more, the better, but you don't always need to have a lot. It just depends, you know.
I had a question. Just you mentioned that it's not known what the mechanism of disease is for the repeat expansions necessarily, but they're common among neurodegenerative or neurological diseases. Is there any ideas about that?
Yeah, so we do know mechanisms of why they can cause disease, but we don't know why it's only neurological disease. So repeat expansions, especially if they're in, well, not really actually. So let me start again. So repeat expansions can cause the protein to not be expressed. They can cause aggregates in the brain, so abnormal protein aggregates. So example, a lot of the ataxias and other diseases like Huntington's disease, the repeat expansion is located in an exon that results in the protein becoming abnormal and leading to abnormal protein deposition in the brain, leading to cell toxicity. So that's another one. With Canvas, we actually still haven't figured it out, and another team discovered Canvas at the same time, and they had a really nice paper in Nature Genetics, and they had access to brain tissue from a person with Canvas, and they did everything they could think of to try to figure out what was the mechanism, and they still haven't figured it out. We assume it's some kind of impact on the way the protein functions, but we still don't know. So mainly gene expression changes, protein changes. You can also actually have RNA toxicity. So if the STI is in an intron, it can become abnormally transcribed. So we call that RAN, RNA. Sorry, I forgot what it stands for. RNA. So it's a type of RNA-mediated toxicity. That RNA itself can cause damage to cells and then cause disease. But why is it only causing neurological disease? That's such an interesting question, and we're trying to figure that out, but we don't really know.
Great, thanks.
Yeah.
Any other questions in the room?
How do you, when you're looking for a new disease, how do you pick a family what makes a good one? Why do you start following someone up?
Oh, what do you mean by new disease?
Well, when there's someone that's, presented with some sort of, well, I guess it's not a new disease, but new for you, why do you choose one to follow up?
So in the context of our research, we know we're looking for ataxia. So anyone with ataxia that we think might be a hereditary or genetic form will be recruited into our program. And then if they have affected family members, we will recruit them. Just the way our study is set up at the moment, where we're going through a lot of clinicians who are not part of our research, so it's a bit difficult to approach family members at the moment. That's why we're primarily just taking the individual patient. But there are other examples, such as the UDP, so undiagnosed disease program, and these are popping up in a lot of countries now, where it's basically large groups recruiting people with unique diseases that they think are genetic, or most definitely are genetic, so really severe syndromic type diseases that occur often in children, and they're recruited into these programs to see if they can find a genetic cause. Those are really difficult because usually it's only one person. Oh, my battery's about to die, okay. So it's usually only in one person, but they would recruit the family, because if it's only in the child and not the parents, you might be looking at something de novo, and then you would want to see what genetic variant is in the child but not in the parents. And that would make it a lot easier in that case if you have that information, if you had the parental DNA as well.
I did have another question about how can you be sure whether the disease you're looking at is monogenic?
Good question. So you can look at the inheritance in the family. If there's 3 generations of people who all have the disease, it is more likely to be monogenic. In the context of ataxia, they are always monogenic. There's no... so people have done GWAS, so looking for polygenic variants that can cause disease, and there's not really many hits for ataxia. Having said that, ataxia is rare and you need a lot of people for GWAS, so it's possible that they may be underpowered. But as part of our ataxia research program, we've recruited so far 170 or so people, and we have been able to provide genetic diagnosis for about 40%. And one of those, I didn't talk about it today, it's SCAR27B, which is another repeat expansion we discovered, and it accounts for about 20% of ataxia cases in our cohort, and also worldwide, actually. So sometimes you do end up going down a rabbit hole trying to figure out something, and it might actually be polygenic, and you just don't really know. So,
yep.
Yeah.
Well, that just about brings us to the end of time. So let's thank again Haloom for talk today.
Thank you.